Okay, welcome to day 2. For those of you online, can you hear me well?
Yes.
Okay, thank you. Yeah, welcome. Yesterday we talked about the concepts of spatial transcriptomes. Today we're going to dive into coding pretty much right away. Just as a reminder and refresher, we talked about regarding cell as a bag of RNAs, and that's what single cell transcriptomics does. So if you have a table of individual cell, each expressing several copies of different genes, you can use it to analyze the cellular properties. But specifically for spatial transcriptomics, we regard cell as a bag of RNA, but in a spatial context. Each cell has its location in place. So in addition to this gene expression table, each cell we measure also their spatial location, X and Y, if it's a 2D slide. Usually most of the spatial transcriptomics at the moment is 2D. There are some 3D that'll try to look at a chunk of tissue rather than the thin slice of tissue as well. So if that's the case, spatial location would be X, Y, and Z, 3D, three-dimensional location of a cell in space. Then we talked about 2 papers, how we can get this information from cells, from a tissue section. So again, today we will talk a little bit still about how to get this table computationally. What are the steps that goes from the experiments raw data to this table? Just as an overview, but we will mostly focus on what to do with it once we have a table, once we have a data set like this. We will be doing 3 coding sessions, making a plot, doing PCA, making a UMAP, and I will give you plenty of time to try and show your results. So first of all, how can we get a table like that from the raw imaging data? If you still remember how we do spatial transcriptomics, the first paper we talked about is called MERFISH, right? They use imaging to hopefully get a lot of gene expression information from many cells. So how is that done? How do you get from raw images to the table, to the cell table? So first of all, they take many different images of the same cells. So this is one example. For the same cell, you have 16 rounds of imaging. Each round is conceptually called 1 bit. You extract 1 bit barcode from each round of imaging. So that you stack them up and make sure which spot is which. Then from there you generate a spot table. Every spot is a single mRNA molecule you detected. A fluorophore, aggregate of fluorophore derived from that molecule. So each spot has a spatial location in this image. We call them X and Y. Usually it's also a stack, an image along the Z direction as well. It has certain depths. So you have X, Y, and Z. And bit 1 to 16 corresponds to whether that spot is on or off in each of those 16 images. Okay, so that's the transcript table. From this binary barcode, bit 1 through 16, 16-digit barcode, you can search your lookup table and see, oh, which gene does it correspond to? Because initially you designed the bark, each gene is associated with a barcode. So you can get from spot, you infer transcripts. So each spot is 1 transcript, and you get to know which gene they are from based on this barcode. But sometimes you need error correction, if you remember. And sometimes it may just be noise, or the error is too much, and so then you just disregard that spot. There's no way. So we call that spot calling, and we call this step decoding. And then comes cell segmentation. So essentially now you have a list of molecules, their place and which gene they came from. you wanted to organize them into individual cells because each cell is a bag of those transcripts. You wanted to assign them to different bags. To understand and to call the boundary of each cell, the territory of each cell in space, you usually do a different staining. A typical one is DAPI staining, which labels the nuclei of cells. Nuclei is the center, is usually in the middle of the cell. It doesn't incorporate everything. So we often also do poly-A staining to get all the cytosolic territories as well. Or people uses different methods to label cell boundaries or just simply aggregate the density of all the transcripts. Usually that also show where roughly where the boundary of a cell is. So once you have those cell boundary or cell segmentation, you can aggregate those information, just count for each cell, their spatial location, and then count how many copies of each gene you measured there in each cell. Okay, so that is essentially a cell matrix we just talked about. From there, we do downstream analysis. So these upstream results, processing, we call imaging processing, data pre-processing steps, are specific to each experimental technique. You designed your gene panel, for example. From that, you know how to decode them. Or if you use a different method, say, sorry, it's not MERFISH, it's it's a different workflow. It's sequencing based workflow, such as slide-seq. Then you will need to have a different processing pipeline. So for slide-seq, because it's sequencing based, there's no raw images involved. And you just have a bunch of sequencing reads, read 1 through n, whatever, that has their full sequence. And we knew from experimental design that part of the sequence came from endogenous mRNA you extracted from tissues and cells. The other part is a barcode that you introduced to encode spatial information. Remember, we pack beads, each with a unique DNA sequence, a short oligo attached to the beads that encodes spatial information. So you parse those two parts out. One part will map to the genome that tells you which gene that sequence belongs to. The other part goes to tell you that came from the beads, will let you to look up their spatial coordinates. So in the end, it's still a counting problem. You get I guess you first get you get a table of beads. Each bead is roughly a cell, but maybe a few cells because of the local diffusions and things. So you can treat it as a sort of like a cell matrix for each cell, but more rigorously for each bead, you have their spatial location and how many copies of each gene you extracted with that bead. Okay, so essentially they all come down to the same thing. Okay, the cell by gene matrix plus spatial location, and that's the table we are going to work with for today and for tomorrow. So we will be doing a few exercises, probably 4 to 5 coding exercises to learn to extract information from this table. Usually, I guess nowadays there are many published studies. This is the starting point people work with the analysis, regardless of what technique you use to generate spatial transcriptomic data. So first of all, we could ask where and in which cell is a particular gene X you're interested in expressed. That's a visualization problem. Just make a plot. So you measured many genes in all these different cells. Each cell has a location. Can we treat it as a staining method if you're interested in the expression distribution within the space for a particular gene? can you make a plot about that from the table? Once you can do that, you can do it for all of the genes in the data. So then next, can we capture the main components of transcriptomic diversity between cells? That we uses dimensionality reduction. So for the first one, for example, if the data set contains 500 genes that's measured, each gene may have a slightly different pattern or completely different expression pattern. But what's the main takeaway in terms of the cellular organization within this data? Can we capture the main components, axes of variation between those cells? That's dimensionality reduction. We can also do, so one way of summarizing the data, reducing the dimension on genes. Another way is we could also think about it as cells. Some cells are more similar to each other than others, and that cells are usually thought of as falling into different types or different groups. Are there different types based on their gene expression information or spatial information? But that's clustering is now a quite standard way of analyzing these kind of data. And once you have cell types information, you could summarize the data in different ways. So we were talking about putting a summary on each individual cell, but you could also summarize data on the other axis and ask for each cell type, what are the aggregated profiles of gene expression? We call that pseudo-bulk gene expression profile. That name came from historical reasons. Before all of the single cell stuff, sequencing are usually done on a bulk tissue with many cells. So the signal you get from gene expression are usually a combination, the aggregates of many single cells. But now we can still do bulk, but we bulk them computationally. So to a certain extent, that's much better than previously. For one thing, you reduce this error if you aggregate a lot of cells together. You will see that. For another, you have full control of which group of cell you wanted to bulk together. So that's much better than traditional way of doing, where you have no control of no way to know whether the things you're grouping together are different or not. So finally, we could spend a bit of time talking about which cell types interact with which other. So this is something new to spatial transcriptomics that's not possible to do with sequencing methods or sequencing omics without spatial context. Because now you have both cell type and know where they are. You could ask, for example, are there certain cell types tend to be interacting with each other more often than expected by chance? Or are certain cell type like to avoid each other or certain things? Okay, so that's an overview that covers today and tomorrow 5 different exercises. So we will be doing a coding boot camp for today and tomorrow. For each exercise, think about it as a workflow, a for loop. We loop between exercises. First, I will show you a demo or some talk, usually me talking about the concepts or show you how to code on the simulated data. And then there will be a coding exercise where you will apply the techniques we learned on some real data I will provide you today. And in the end, you will show your results in a shared Google Slides. Again, I will show you how you can do that. Again, pause any time for questions. Okay. So let's jump right into the first coding exercise. It's also the most straightforward conceptually, but if you haven't done coding much before, you will find there's still some chances to learn about this exercise. If you feel like this is too easy to you, you can ignore this and go straight on the bonus exercise 1. I won't talk about it. You will find it. Okay, so essentially you will just practice data visualization on a public mouse brain dataset. from Merfish, provided by this company called VisGen, and that was made public. So what you wanted to do is to make a scatter plot from scratch. I will give you the data, but then you make a scatter plot from it, trying to show the location of each cell, and then plot the expression level of one gene color coded onto this. Yeah.
Do you have the material somewhere?
Yeah, I will. Yeah.
Okay.
Next slide. Next
Okay.
slide. Just to give you, yeah, great. So usually the first try you will get is from you will make something like this, which seems to make sense. It has some progress, but not sure what we are getting. In the end, you will get to some place like that on the right, and you will figure out some nuances doing that. Okay, so I provided you with a coding environment, so you don't need to set up anything. You just need to go to this link, and then it's a GitHub repository. Many of you might be familiar, might not be familiar with it, but it's how we store our code and share our code. And then inside the GitHub page, just open workbook. There's a folder called workbook, and then open exercise 1, then click on that icon. That'll bring you to a coding environment online. Yes.
You sure it's that one?
Yes. Sorry, I haven't shown. Let me show you the link. Let me send the link to your email probably.
I could type it if for just in case I use it or something.
Okay. Sure. After class, I'll definitely provide all the links and some materials and solutions, and yeah, no worries. Yes. So if you don't feel like you follow up along, we can slow down, and also don't worry, I will provide all the class material solutions tomorrow and along with your homework assignment. Okay. So yeah, let me give you 5 minutes to set up, and probably after 5 minutes, I will show you how to set it up in case you hasn't. So then we can all be on the same page. Okay. Yes.
Sorry, I have a question that, I mean, it might sound silly, but it's not super clear to me. So right now, it is that we have an outcome at table with X and Y and the expression of the genes, so the different reads. What is not clear to me is that this I can picture it as coming out from the analysis of one tissue section. What is the output when I send replicates or even better biological replicates which can have bigger variation, right? So do I get a consensus table in output or?
Yeah, the raw data will still be, for each section, you will have a set of table like this.
Okay.
Then you begin to worry about whether how to integrate them together, right? And so, for example, the raw X and Y location wouldn't make sense when you just simply put them together, because one section is over here on the micro, the other is over there.
Yeah.
So you need to rotate them and find the common, spatial, the true spatial relationship that makes sense to you, yeah.
So I will run different analysis and then the interpretation of the biological meaning will come like later.
Will come later.
Each of them.
Right. Or you could try to dive into each of them separately and see, are there any aspects of them consistent or yeah. So
Okay.
yeah.
So have pipelines to pull different replicas.
Are there pipelines to pull different replicates together? Yes, there are many different tools to do that. But you need to be careful though. You need to understand what you're applying, right? And say, what do you mean by pull different replicates? I guess you usually wanted to worry about two things. What does it do to the gene expression level for each data set, right? Do you understand that part? Another part is what does it do to the spatial part, right? You know how you collected two sections, their spatial relationship. They usually don't know. So you need to specify how you want to combine the spatial information. Otherwise, it doesn't make any sense. There's no way they figure out. So I guess there may be ways if you provide them with a mouth atlas and you want them to register them onto a general framework, then you know which slide is within the, but there will be a lot of back and forth when you do those kinds. Yeah, great questions. Again, as a reminder, if you're online, you can type out your question in the chat, or also just unmute yourself and raise your question. Okay, make sure you give breaks every hour. I should have. Yeah, we did that yesterday, right? So today will be shorter, hopefully. Okay, let's try to do it together if you haven't got to set up your coding environment. So first, let's just go to this link. Let me share my full desktop. Okay, so this is the GitHub repository that we are going to work with. It actually already has the slides from previous sessions and so on. So I will be using this link as well. So so here is a Google Slides we can use in class. Let's open that. This has all the coding exercises and data stuff we did from previous sections by everyone. So let me just get rid of all of these. Oops. So, and adding some blank spaces. So we will be sharing our results over here in a minute. Okay. So let's go to workbook and then exercise 1. Here's bonus exercise 1 if you're interested. So let's go to exercise 1 and click on this icon open in Colab, that brings you directly to a computational environment provided by Google Collaboratory, which is also a collaboratory. So you can immediately just run these code. Exercise 1, here's some introduction, how to make a scatter plot and iteratively make it better. So here is a section of the code. If you are not familiar with Python, you can run it the way you can click this to run it, or you can just type on your keyboard Shift and return. Oh, then initially there's a warning, I'll run anyway. So just Shift and return, it'll run this section. So it says, so now it has a mark that indicates it has been run in 0 seconds, so instantly. So the thing we did here is import some packages that you need to use to do this exercise. So similarly, you can run the next code sections. That's just a function I defined for you. You don't need to worry anything about that yet. Just run it through. Okay. So first of all, let's make a very basic bare bone scatter plot. What do you need to make a scatter plot? You need to specify the location of a few dots. For each dot, you wanted to let the program knows their X and Y location. So that's how you do that. You have a bunch of X coordinates and Y coordinates, and then you use one line of code called pld.scatter, sending the information of X and Y, and just run that, you generated a scatter plot. Okay, so that's how you generate a scatter plot using X and Y information. 3 lines of code. Specify X, specify Y, make a scatter plot. Okay. Now you also wanted to color it by a gene. So I'll leave that to you to figure out how you can color the dots by a different color specified by a different value, right? But let's jump into the real data. So here, in this section, I provided you link to a real data, and this code will just retrieve data from that link and present you with a data table. And that information is stored in a variable called data. So if you just run that, it takes a few seconds because the data is quite large. So now you see this data table is here for you to play with. Here's some information about this data table. Okay, so it says it has 84,000 rows, 485 columns. What does it mean? 84,000 rows. Each row is a cell, okay, being captured by spatial transcriptomics. So in total, you have 84,000 cells in this data set. What does 485 columns mean? So you could go up and see the column names. Most of them are gene names. So you measured 480 genes or so. But the first 2 columns are X and Y. They're spatial locations for each cell. So these 2 different bits of information. For gene names, there you see a lot of zeros. Are they all 0? No, they're not all 0. For example, here this cell has 4, this cell has 2, but quite a lot of 0. It's because most of genes are not expressed in most of cells, or you didn't capture it. And you also notice that for genes, it's always an integer. Why is it an integer? 4, 2, 5, 3. That's because it's just a counting problem. You count how many genes have been identified for that cell. It's always an integer to begin with. Whereas here for X and Y location, they are not integer. They're just continuously, it's continuously based. Okay, so that's your data table. Now your task is so now you have the data table, X, Y location for so many cells, and many different genes. You just pick one gene. If you don't know which one to pick, I suggest you go this gene, SLC17A7. That's one of the genes in the table, but you could also choose any genes you wanted to do. So your goal, is that clear? Your goal is to make a scatter plot to show the location for each, for all of these cells, 84,000 cells, colored by the expression level of one gene, SLC787. So I'll just leave it to you. There are, I guess, let's do it. So for the next 10 minutes or so, you can play with it yourself, and maybe you can spend another 5 minutes, talk to your neighbors and see where you're stuck, or just learn from each other. And after that, we'll do it all together and address your questions. And I will be going around. If you have specific questions, I can help you. Okay. So
Can you show the example that we kind of skipped, that you can work on it together, the iteration?
You mean
Two.
this one? Iteration 2. I skipped. Yeah, how do we make a plot colored by a gene, right? So you need X and Y and gene. So let me just show you this actually. Now, we have AI tools in our disposal. Let's ask this question to ChatGPT or whatever AI tools. Here is Cloud, but you can use ChatGPT. Okay, so you just ask make a scatter plot in Python, color it by some different values. Let's see how they okay. Says I'll create a Python scatter plot with color coded points based on different categories. And okay, so yeah, they generated a lot of code. Let's just try to run it. Just copy it and run it. Without looking into what they says, let's first try to see if try to check AI if it's correct or not. Oh, now they made a scatter plot for you, right? I just copied all of their code, and it looks like they made a scatter plot colored by 3 different colors. I didn't read through their code; it's quite complicated, but that made some progress. So if you were able to do that, you can then go back to the code and see which line actually did it. But in this case, though, I knew it wasn't what we really wanted. For why is that? Because gene expression is continuous. Here, they seem to be making a plot that show you 3 categories, 3 predefined categories. It's not quite what we want, but we can let them to change, right? So that's not really what we wanted. So we could say I wanted to color it by some continuous value, okay, rather than discrete values, 3 categories. That code, though, will be helpful if you have 3 cell types you want to color them that way, right? So let's see. Okay, now they made a colored version of this code. Again, without seeing what, reading through what it says, let's just test, copy and paste it over. Now, yeah, this time it seems to do what we want, right? It's making a scatter plot colored by a continuous value that goes from maybe 0 to 3.5. They that makes sense. It's a bit chaotic, but I hope to convey the message that you can ask your question to AI. You copy the code they generated to your place and see if it works. If it works, that's a big progress. Then you can go in and understand which line actually does the work. Okay, then you can maybe learn and adapt from it. But don't worry, I'll give you plenty of time to try it, and I'll go around for questions. And after 15 minutes or so, we will review it together. Also, you can use ChatGPT, ChatGPT, Claude, or Perplexity. All of these should do the work. AI coding assistant. Yes.
I undo something? Can I ask you this one?
Oh, how can you undo something?
Yeah, if I wanna undo.
Ah,
This one, it says. I really don't.
maybe.
I
No, yeah. I guess you can copy from your from. Oh, yeah, question. Yeah, yeah. I forgot to mention it, sorry. Yeah, we can do it together too. So let me just show you how you can do that. So say if you get a plot like this, you could, I don't know if this works. Let me try. So if you right click and copy the image, and then you go over to the shared Google Slides, then perhaps you can paste it. Yeah, then yeah, you can write your name down or whatever. This is my plot from main, and you can create a slide for you. Yeah, that's another way is if you, I believe you can just drag things over to, right? It's too crowded for me. But if you say put your slides on this side, okay. Anyway, so here's your plot. If you wanted to drag it over to the Google Slides, you just move it like that. And then, yeah. The link is in the GitHub repository, the homepage, and then you scroll down. I just sent the link to the repository to your email just now. We will be using this link over and over again for today and tomorrow. And I'll send the link to this slides as well. Let's see. Okay, how was it? If you have any, if you have some question, you can talk to your neighbors, share what you got. And Okay, let's try to go over this a little bit together. Yeah. So yeah, it looks like many of you have made a lot of progress. Wow. Yeah, good job. And anyway, let's do it together. So I will be coding right here. So hopefully I just show you one technique, maybe one now the most important technique how to code. One is to know you can type, and the other is to know how to raise a question to AI chat box and then copy and paste the answer to you, and you can verify and tweak with it. The 3 years ago when this class was started, ChatGPT hasn't came out, so we wasn't coding this way. Apparently over the last 2 years, things has changed a lot. And this new way of coding is called vibe coding or whatever. But I feel like still we need to understand some of the basics. So AI can help you go a long way without knowing what to do much of things. But at the end of the day, it's still better if you can use it to teach you what are the key steps and key ingredients you need to accomplish what you wanted to do. Okay, so let's take a probably minimalist approach again from here. So in order to make a scatter plot, you need a list of X and Y's, and then that made it for you. That has made it for you. So next, I wanted to add an element, which is to color each dot by a different value, right? How do we do that? Apparently, we need a third information called gene. This is just some dumb examples. Gene value 0012, 0 copies of this gene for the first cell, 0 copy for the second cell, 1 copy for the third cell, and so on. So once you have that information, how do you put it in here? Let's say you didn't know how to put it here. Traditionally, you could search Google and look up for the document for this particular function people provided for you and see is there an option for you to add a coloring step. But now with AI, what we usually do is to ask them to accomplish this for you first. So now it's already done. Then you look into the code and see what ingredients is key in setting things up. For example, you see there are so many lines of code. Part of it is just to generate some random data for you to play with. So let's ignore that. Part of it is to say label things, show there's a title and stuff. Let's ignore that for now. The key line is still just this one line. Okay, plt.scatter, which is the same code we used before to generate XY coordinates. But see, now they provided many different things, different options to enrich that line. So you could guess which line actually did the work or which option. So for example, alpha, I don't know what alpha is 0.7 means. Edge color probably is for some decoration. Line width, I don't worry about that for now. But the key one is this color values, right? C equals color values seems to be the key. So C, color values is just what they generated. They generated a random data called colored values, and they use that to make the plot. So the essence, the key is to know that you need an option called C equals something. Then you send in the genes version value. Okay, so let's go back to our minimalist code. So the key step here is to add this option called C equals the values you want to be colored with. So in my case, it's called gene because yeah, or you can call it whatever. Let's call it colors, maybe to be. So then if it's colors, then colors here. Okay. Let's see if it made a difference already, right? And because that's my first cell, second cell, third cell, fourth cell. The fourth cell has the highest level of gene expression in my toy model, right? And then so they are represented by different colors. Okay, so now you learned in order to color it by something, the option is C equals something. Once you figure this out, you can use it to to generate many different things. Now let's apply this code to the actual data we have. Okay, let's skip ahead. We already loaded the data. So now let's make a plot with the actual data. Now we just need to redefine our x, right? Our x is now from the data structure, the data table, the way you can, so I just do this, x equals data, and you select the column x, y equals data, and you select the column y. And you wanted to color it by some gene expression level. So gene expression equals data, and you select a particular gene. For me, I will select SLC17A7. So you do this, scatter plot X, Y, and gene expression, okay? So then you run it. So you might ask, oh, I don't know how to select a column from the data. Why is that a square bracket? Why is that a quote? Don't worry about it. Just first ask that question in ChatGPT, right? And then let's try to do it. How to select a column from a data table in Python. Okay, and let's see what it does. Okay, step 1, import pandas is a package. I already did that for you. So step 2, create a data frame. You already have a data frame. Okay, step 3, select a column. That's the one you need. It turns out the syntax you need is data table, square bracket, a quote, and column name. That's the one I just used. I knew that already. So here I don't need to consult my AI chatbots, but if you need, just do it like that. Okay, and then make a scatter plot. It take a while to do that. I will. Okay, 7 seconds to make it. Why is that? Because now you have many cells. Okay, that's where that's how we get to this point. Okay, so that's a mouse brain section with many cells, and this mouse brain section is a little tilted towards the right, right? And then it's colored by a gene, but you don't really see much pattern, right? What's going on here? So one issue is that I guess you have too many cells, right? Each cell is quite big, and there's no space to put in so many cells without being too crowded. Okay, so if you figure the problem is you have too many cells and things are too crowded, you may want to say shrink your my scatter plot dot size or the point size, and ask how you can do that. Okay. So I just asked the question, can you shrink my scatter plot point size? Because my worry was we have too many cells on top of each other, so you don't see the structures at all, right? So here's an example ChatGPT provided for you. So by default, it's a large point. Why is it a large point? Because it's s equals 100. So there's something called x equals 100 that may be related to the size of your point, right? And for a smaller point, you do s equals 20. So yeah, that seems to make a difference, right? So first of all, without doing anything else, let's just copy that code over and see if it makes a difference, right? And you can run that code in here. See a larger point in blue, a smaller point in red. That's the code the AI provided me with. So then you can play with it. Say, oh, what if it looks like the key argument here is to change this s equals 20. If you can change it, say, into a smaller number, what if we do s equals 1, right? Sorry. For some reason, my code just stuck. Okay. It's lagging for some reason. So what if I just put s equals 1 rather than s equals 20? Would I get smaller dots? Yes, I do seem to get much smaller dots. Okay, now you've learned that. You can use it to change your previous plots, right? So now, previously we had this big plot x, y, and c, very clean. Now we just need to change it a little bit, right? Add the argument called s. Let's try it. S equals 1. Would it make a difference? Yeah, it does seem to make a difference, right? And you could go further, s equals 0.1. That seems to make a difference too. What if s equals 0.01? Yeah, you see the brain structures reveal themselves if you happen to know mouse anatomy, that the hippocampus is very obvious over there. But there's still one more problem, right? So it looks like all the cells are pretty in this navy purple color without much variation, right? So what's going on here? So is that the gene expression value you wanted to color things with seems to be not normally behaving. You probably have a few extreme values. For example, the gene expression. If there's only a few cells that has extremely high values of expression, but most others have smaller values, and then you won't be able to see the patterns of the variation among the majority of cells. So how do you know that though? One thing is to make a sketch, to do some diagnostic things and see, make a scatter plot, say, this is my gene expression values. Okay, make a histogram of gene expression value and see what it looks like. You don't have to learn this, just to show just to show you what this gene expression histogram looks like. See, so this is the expression level of this gene SLC17A7, and how many cells are expressed with 1 copies of it, 2 copies of it, up to 100 and 500. So you see this is a very long-tailed distribution, meaning a lot of cells do not express this genes, while as maybe a few cells expresses 500 copies of that genes, while some other cells express mostly 100 copies of genes, right? It's very screwed. So if you plot a screwed distribution like that, you will see something like this. There are probably a few cells in here you won't notice that has high value and so on. So how do we deal with that? One thing you can consider doing is to make sure that you cap the value you wanted to plot. Say, if you only wanted to care the range between 0 to 100, because that seems to be where most of the cells live, you can color map only up to 100. And if a cell goes beyond 100, you still color it by the color of 100. Okay, so how do you do that? You can have another argument called Vmax, which controls the maximum value you wanted to color code it with. So let's put in 100, because that seems to be reasonable. Okay. You see that makes a big difference. Now you see a ring of cells expressing high levels of this gene in orange, where the rest of the brain in the middle do not express this gene as much. Okay, so yeah, that's pretty much how you do plotting and tweak with it, understand where the problem is. Okay? So in the end though, let's just review. You need, as a minimum, you need to figure out what you want to do with the cell. You want to have an X coordinate, Y coordinate, right? Gene expression values, and some tweaks on the elements of the plot that make sense. For example, make the cell size small enough because there are many cells, and make the values of the data plotting range useful because given the genes at your disposal. So and then once you figure that out, it's usually just one line of code in plotting. You're sending all the information, figure out which argument to use, and then you get a plot like this. Okay. And then you may worry about, what about all the decoration? Can I add a title? Can I add a sidebar showing the color map and all these things? AI will do those for you. Okay. You just ask. So for example, I guess in that plot, we just ask them to generate, they already provide you with a color bar and everything. You just go figure out which line controls the color bar, which line controls the title, and so on, right? And if you're unsure, you can always ask them back and forth. Okay, that's the first coding exercise. Let's take a break, take a 5-minute break, maybe, and come back at 10, 20, 21, maybe 22. And we will head out to the next session, PCA and UMAP.
It looks like my Google Drive is completely full. Is there another way to save your work?
Oh,
on the
save.
Google?
You wanted to save it, yeah.
Yeah, I can leave it open and then.
I
read it
guess you
later.
can download.
Okay,
Yeah.
okay, well, then I just leave it open, but my Google Drive is completely full. Okay.
Oh, share the recording, guys.
Would that be fun?
Yes, actually, I don't have the recording. Could you send Eloy an email? I think, yeah, if he has it, then we can reply back.
That's okay.
Yeah, definitely, yeah, we always. Thank you.
Sorry, so I
Yeah.
was able to get out of the Zoom today, so I can stay the whole day. Tomorrow I have a long meeting 11,
Yeah,
9 to
no.
11, so I have to skip the
Yeah,
whole day.
no worries, yeah.
But you will probably send whatever
Yeah,
material.
I'll send
Okay.
the material. I won't have the recording, but Eloy will have.
Okay.
Yes, I will.
Okay, thank you.
Yeah, no worries. But all of these is already
Yeah,
there.
yeah,
Yeah,
sure.
all the class material are in that link already.
Okay.
So yeah.
Okay, good.
Okay, let's move on to our next session. Before we do that, let's just review what we just did, right? So the nice thing about coding, you may take a while to figure it out how to do even a simple, say, plotting things on a gene that seems so simple, but you may take a while to do it. But you just need to figure it out for once and for all. So if you figure out how to do it with one gene, one data set, you automatically know how to do it for all genes in all data sets effortlessly. Next time you just copy and paste the same code. You wanted to organize your code a little bit, then next time you just apply it. For example, we just did SLC70A7, right? Then you can easily use it to do a different gene A6. That's a different pattern than GAT1. There's a different pattern. GFAP, yet another different pattern. So you can use it to explore whatever gene you want in that data set. And that's generally true for our coding. That gives you a lot of leverage. So if you just figure out one thing, you wanted to think about how to use it so that you can let computer work for you and apply it to all different things you want to see. So next session, we're going to talk about dimensionality reduction, because each gene is different. There are so many different genes. What's the essence of the structure of that data? So dimensionality reduction says that if you have a big table like we do, of n samples, n is the number of cells in our case, p features, p is the number of genes. Can we reduce this bigger table to a smaller one into the same number of cells, but fewer components, fewer features? Instead of hundreds of genes that you measured, can you reduce it down to, say, 2 components so you can play with it and visualize it? And in the context of single-cell RNA-seq, and it could be hundreds of thousands to million cells. In our case, our data set has 80,000 cells, right? P 10,000, 50,000 genes. In our case, 5,000 genes, 500 genes or so. And can we reduce it down to 10 to 100 components or even 2 components rather than that many genes? Why can we why can we do this data reduction though? Why is that meaningful? Sorry, I want to. That's because of two assumptions. One, maybe the same assumption expressed in different languages. One is that biology always has structure. That means that your table is not random. If the table is completely random, you cannot reduce information from hundreds of columns to a few columns. But because gene has structure, meaning different genes co-express to form functional group, they coordinate with each other. So their patterns will be similar. Some genes will have similar pattern in certain cells. That allows you to reduce and combine them into a component without losing much of the information from the data structure perspective. For data in the data science language or whatever, computer language, it says data often lives in a high dimensional space to begin with. By high dimensional meaning the dimension of your feature, P. If you have 500 genes, the data of a cell can be regarded as a point in that 5-dimensional space, 500-dimensional space. But they don't occupy all the regions within that high dimensional space. They live in a very low dimension, embedded in a high dimensional space. And the key is for us to figure out where they actually are within that all the possibilities, right? So all of these is quite abstract. Let's take an example, right? A toy example. Here we have a data table. It's not quite big, but we want to reduce it. This table has, I don't know, 20 cells. Each row is a cell, right? And 2 genes. Each column is a gene. And if you plot it geometrically, this cells lives in a 2-dimensional space, where x1 shows you the expression of gene 1, x2 shows you the expression of gene 2. If you think 2-dimension is too much, we wanted to reduce it down to only 1 dimension. How would we be able to do it, and does it still make sense, right? So in this case, our intuition can guide us, right? So intuitively, maybe we can, if you force me to reduce dimension by 1, we just get rid of the second column. Why is the second column but not the first column? Because, cells are more variable. You can distinguish more things with the first gene, whereas the second column doesn't seem to contribute as much, right? So in blue is the raw data, in gray is the reduced data. They all fall into this line, 1-dimensional line. And so in this way, we achieved data or dimensionality reduction, right? And by losing some, but not much from the information, removing the low variance column in technical language. Okay, so what about a more complex case, right? What if you have a structure that looks like this? X and one is tilted. How about now? Do we still remove one of the column like this? I guess you can, but it's not, we know something intuitively think we can do better than just dropping one column like this. So what we can do is actually think of it as a rotation. Rather than drop directly one column, you first rotate the structure along some axes to capture the most variable axes in your data. That's one thing. And then you drop, and then you just project all the data on that line and dropping the direction perpendicular to that line. So that's a better strategy that preserves more structures from your data. So initially you had 2 columns. Now you only have 1 line. By using this 1 line, you can pretty much still reconstruct a lot from the original 2 column. So to codify it, we wanted to remove a column, but only after a rotation. And hopefully we did a rotation we figured out a better the best rotation to do in order to preserve as much structure as possible. Okay, so that's the idea behind PCA, and that's actually what PCA does exactly. As PCA reduces data dimension by removing a column, but only after a rotation. But which rotation does it do? It figures out the best rotation for you, identify the best basis in the feature space. And it's all sorted out a century ago when people studying linear algebra. They discovered the process called singular value decomposition, and which says if you have a big table of X, you can reduce it down to and think of it as a combination of three smaller tables, three smaller matrices, and decomposed this way, component by component. The sum of all the components will reconstruct your original table, where these components each has a basis or the axis along which your data lives, and a score. So each data has a score of where they are along these axes, and also how important that component is. And you can rank components by the most important one to the least important one. And that's controlled by a single value called standard deviation. That's literally the standard deviation along the axis of that component. So once you decompose data this way, a big data table can be peeled apart into components, then you can choose to remove less important components. So that still gives you a lot back. And it works in more than 2D. I showed you the example in 2D. It works in more than 2D. Okay, examples in real data. So single cell transcriptomics data have many genes. And if you plot cells in any pair of genes, they look differently. Say, here's 3 examples. These 2 genes, cells look like this. If you choose a set of different genes, they look differently and so on. But you can't exhaust all the pairs, right? What you do, but you can do is you plot all the cells along the first two principal components. Then it automatically shows you the dominant structure among your cells. In this case, we showed you it's a data set of the mouse cortex where we know the cells their properties vary along the layers of the cortex, and that structure is automatically shown in the first 2 PC cells goes along a gradient continuously. Whereas if you choose a pair of genes, you need to figure out what you're looking at, right? And you need to figure out the right genes and maybe the right combinations of them. Okay, so that's one use case. Another use case is it can help you do a better rotation, right? So remember the data set we played with is this. The cell locations and spatial locations are tilted. That probably is from the way people initially use imaging to in the imaging setting, the samples are tilted or whatever. But if you want to rotate your brain slices back to its normal position, like this, you could try rotate it differently and figure out, but you could also apply PCA because PCA does nothing but rotation. If you have 2D data to begin with, it will send you back 2-dimensional data, but after a rotation. In this case, they will always find an axis that along which the data has the most variation. So it turns out the normal position along left to right cells have most variation, right? So if you apply PCA to all these brain slices tilted in different ways, their PCAs will always be something like this, more square like this. Okay, so let me show you PCA and you will apply PCA to the data we just had. I wanted to show you PCA on the toy data. So I will generate a simulated data like this. I have a bunch of different cells. Each dot is a cell. They live on a ring structure. The ring has an opening in order for you to see what PCA does to that ring, right? So I wanted to apply PCA to this two-dimensional space. You can think of it as gene 1 and gene 2. Cells are cycling or whatever. After applying PCA, I wanted to show you PC1 versus PC2. You will see that what PCA does is nothing but a rotation of your data. But they always rotate your data however they originally oriented in a particular way, such that the first axis always capture the most variance of your data. Okay. Then you can do dimensionality reduction. Say you remove the second component or whatever. Okay. So you can open. I guess you can hold it off. I'll give you time to open exercise 2, but I just wanted to show you how you can generate everything perhaps using AI. Let's challenge it. Okay. So first of all, I wanted to generate a ring, make a scatter plot of a ring, maybe. Let's see. Ring-shaped scatter plot. It's generating some code. Once it's finished, let me just copy this code entirely and see. Looks like it did the job, right? It generated a ring for me. It produced some simulated data, but that's not quite what I wanted, because if it's a ring, you do a rotation on the ring, you wouldn't notice it. So I want the ring to have an opening. So maybe I will let the actually I could let AI to do it, but I could also go in and look at the code and see what it does. Okay. So theta it looks like theta is an angle they wanted to have that goes from 0 to 2 pi, and with endpoint, n seems to be 500. They generated 500 for me that lives on the theta angle, right? So if you go all the way from 0 to pi, you know it's a complete ring. But I probably don't want it to go from 0 to 2 pi. I want it to go from maybe 0 to 2 pi, but times 90%, say 90% of 2 pi, so that'll create an opening. And see, if I understood the code correctly, I should generate a result, right? Okay, so that is an opening. So that's something I can work with. Then I just wanted to apply PCA to this data, right? So then we can go back. Say if I don't know how to do PCA, I wanted to do PCA on this data. Okay, so it understood that PCA means principal component analysis, and I already have the data and so on. It generated some code. Again, I can copy the code entirely and see if it works. But at this point, I already trusted AI so much that I think it will work. But I actually just going to go in and see which line in particular does the job, right? It looks like here is generate ring data. It says generate ring data. I'm already familiar with, and I already changed it to generate a ring with an opening. Then I only need to focus on this, apply PCA. So actually, I just wanted to copy this bit over here. Okay, apply PCA with X. Let's see. Oh, once I apply it, it doesn't work. Why does it not working? Let's read this error message, right? It says name error, name X is not defined. that's a simple error we often make. That just means you wanted to apply PCA on this data X, but you hasn't specified X yet, right? So let's go back. How was X specified? It looks like we need to also pay attention to this code. X equals this, because we had X and Y coordinates. We use it to make a ring, but we also wanted to combine them and send it into PCA. And that's where X is defined. So I left out something. So let's copy it over that line of code. X equals column stack, meaning stacking two column data together, right? Okay, it looks like it went through. Okay, so if you check, one thing we always wanted to check is to always check your results. And in my case, the results is called X_PCA. And when you're working with big tables, the first thing you want to check is what's the shape of that table? How many rows, how many columns, right? Let's do, oh, sorry, not reshape. I will do shape. So it shows you that you have 500 rows and 2 columns. Why is it 500 rows? Because in the simulated data, they generated 500 cells for me on the ring. Why is it 2 column? Because we have 2 features, X and Y, right? Okay, then I just wanted to plot this thing, X_X, X_PCA. So it looks like, though, it's already provided code for me to plot the data. So it says plot the PCA results. You took the results as X_PCA, and what this thing does is you select the first column from that X_PCA. You remember X_PCA is a 500 row by 2 column matrix. Here you select the first column, here you select the second column, and so on, and put on some labels. So I'll just use that code and copy it over. Okay, now is the PCA transformed ring data. Yeah, they said it exactly right. So this was the ring-shaped scatter plot with X and Y raw data. After applying PCA, we visualize PC1 versus PC2, and what it does is it moves the opening to the upwards to the Y along the Y direction. Why is that? Because in PCA, the first PC always captures the most variation. So if you have a ring, the opening can't be along the first PC. It has to be perpendicular to the first PC. Okay, if that makes sense to you. It might take a while for you to understand what that's okay, but hopefully at least I show you how you can apply PCA, right? The key is, so for your exercise, you already have the data. It's higher dimensional, but the key is to do these two code. You figure out the X, you send it into PCA with how many components you have, and you visualize the results. Okay, so that's exercise number 2. Yeah, go ahead and can do exercise 2A. You will be applying PCA to real data. The real data has more columns, but you already know how to get the real data, and I already provided code for you. Just open exercise 2 and 3 in the GitHub repository, and do PCA and plot the first 2 PCAs. Again, once you've done that, or by the end of 10 minutes, you can share your results over to the shared Google Slides. For example, I will just share this ring data. I'll copy that ring data over to my space. That's my result. Okay, 10:45 now. Let's spend 10 minutes, 10:55 to do this analysis. And I will go around for questions. So if you think you're stuck at some point, let me know. Okay. Okay, so in order to run the first two sections, and it tries to import and download some data packages called UMAP. That will be used later, so it might take a few minutes for it to download. Just I guess you just need to wait a few minutes. It'll do it afterwards. It'll be easier. Is the sign-in sheet somewhere? Okay, okay, let's do it a little bit together. Okay. Looks like so how many of you have gotten to the point where you have loaded data in exercise 2 and 3? You have a data table to work with like this. How many of you have loaded data? Yuri? Okay. How many of you have done PCA and plotted the first two components? Okay, just a few. How many of you have just done PCA but couldn't figure out what's going on there? Okay, a few. Sure. Let's start from the data table. Looks like all of you or most of you have gotten to this point having a data table. So the first thing we did is a code I provided you here called I selected the data table but excluded the first two columns. This iloc meaning selecting according to the location of this data. First, I selected all the rows, so before the first comma. Then I selected pretty much all the column, but excluding the first two. So I selected from the third, I guess, including first two, and all the way to the end. Why do I need to do that? Because the first two columns are just spatial locations. They're different from gene expression. So in reducing dimension, we're talking about reducing the dimension of in the gene expression space. So I just essentially just subselecting the data table to only only include the gene expression data. Okay. So then this is the table we will be working with. It has 483 columns. The goal is to reduce it down to 2 or 20 columns with the same number of cells. And that is done here using PCA. Here I provided you with some templates you can play with, but this is all because for historical reasons, when the class started, the standard way of learning how to code is to go read the official document of this package. So I provided you a link to the document. It says everything about how you can apply PCA and so on. But now you know you can use AI to generate an example and you can understand the example. Anyway, so I will just go ahead and do it because I already know the syntax to do this. So in PCA, there's a column argument called n components, and it specifies how many PCs you want to get from your data. Let's say 20 PCs to begin with. And then once you define that, you wanted to send in your data. In our case, it's X. Sorry, it's not X. It's called features. Features. That's our data. And then out you produce the PC components. And it's always good to check the results by its shape. Okay, so let's just run that PCA. Okay, so it says it takes 2 seconds to run because I told you to keep track of time. But also this print shape says the PCs have this many column and this many rows and 20 column. That's exactly what we needed, right? That's 20 PC components. Then
So what's the time about? What's that?
this is a little decoration telling it to keep track of the time. So you return. So if you wanted to run a long code, you wanted to get a coffee in between, say, you wanted to keep track of time the next time you have an expectation of how long it takes, right? And for a shorter term code, making a plot, you don't want to do that. So if you don't include that, it will get, if you comment that out, it will still do the PCA, but without showing you time or anything. Okay. Okay. So that's the piece we've done PCA. So now we can make a plot of PC1 and PC2, color it by the expression level of this gene. Actually, I provided code here, right? So if you just run this code, it shows it. Right? So PC1 is the first column of this PCs. PC2 is the second column. We generated 20 columns, if you remember, 20 PCs. So that's that. And gene expression by SLC17A7. Here I did a log transformation. That's also a common thing to do. If you remember, if you just plot the expression level of SLC17A7, there's a few outlier cells that express 500 copies of that genes, while most of it is below 100. So this log transformation helps you to review data in a more balanced way. You can also not do that for the sake of PCA. And then I make a plot showing. So the plot, we just learned how to make a plot. Here, I just summarized it as a function provided to you. So that's it, PC1 and PC2 colored by this gene, okay? That's that. Let's go back to our slides and see that, yeah, it's the same result. So what can you learn from this? What does PCA do to the actual data, right? It's like a flow cytometry, if you want, where you can see cells can be separated into different branches. A branch goes like this, a branch goes like this, and a branch goes like this. This gene happens to label two of the branch. So cells that are on this, or you think of it this way, cells that has relatively high value of PC1, who are located as over here, also are labeled by this gene. So this gene explains part of the PC1 or contributes to it, right? So for a certain cell population. Okay, so yeah, that's PCA. Let's take another 5-minute break. Let me know if you have any question, and we will do our last stretch doing UMAP.
Can you display the last PCA?
Yes, let me go back.
The plotting?
Yes, this one, right?
Yeah,
Yeah.
thanks.
Okay, okay, let's do our last stretch. But let me just highlight one plot by Cassie. Thanks for doing this. And this is also part of the plot people often use to show PCA, right? So it shows as the number of PCs increase, what are the variants explained by each of the components? See, it always decreases because PC1 often definitely explains most of the variance in your data. Here it's a bit more than 0.4. What does it mean? It means 40% of the total variance in your data can be captured by first PC1 alone. And then the second PC captures another 20% or so variance. So all of these PC adds up should equals to 1, because in total you have, think of it as a pie that's 100%. PC1 take over 40%. PC2 take over another 20%. Remember, in the SVD, you have a big data matrix, which can be break into component. One USV, that's just some code. But each component, this whole component is called PC1, which accounts for 40%. Then if you add up second PC, counts for another 20%, so on and so forth, and then you get diminished return from that. Okay, that's how PCA works, right? By breaking apart these complex things into manageable components that can be added back up together. Okay, so yeah, let's move on to this. Actually, in order to motivate the UMAP, let's discuss when does PCA fails. Okay, let me provide you with a few examples. When does PCA fails? If you, I show you, if you have a ring, it rotates a ring. It means it doesn't do, in the context of dimensionality reduction point of view, it doesn't do much. Why is that? Because we know in this case, the data lives on one dimension. It's just a ring. It can be represented by an angle. Say if the cell is cycling, its cell is living along a trajectory of this ring. But PCA will have no way of capturing a ring in one dimension because it only do rotation. It doesn't squash the data at all, right? So linear rotation versus nonlinear transformation. In order to capture a ring, you needed to and press it down to one dimension. You need to open it up and press it down to one. So that involves a nonlinear transformation. Also, dense component versus sparse modules. We often wonder about. We also wanted to think of gene as acting together to form modules doing similar functions. But PCA doesn't really do that. Say, for example, you can't interpret PCA as a module of genes. There are genes contributed higher to PC1, for example, and those that contribute less. But pretty much all genes contribute to all PC components. That's called dense component, meaning yeah, it's not just a few genes that contribute to any PC. They doesn't care about this module structure at all. And in the end, also this is a problem for all the data analysis. Most of the data analysis, you need to make sure what you're doing and know the limit, right? It's always a correlation, not a causation. It's just the data in the table, and you can generate a lot of different patterns with it. It doesn't necessarily mean much unless you understand what's behind the scene, right? I guess in this image, if you think about yourself as an alien, you came to the human society and you don't know why the truck is moving. Is it because there's a driver inside paddling, or is it because this person is pushing? Is it because this person on the truck is pushing, right? All of these are correlated, but which one is causing the issue? Biology knew how to do it, right? You knock out one person at a time. and figure out when does the truck stop moving, right? So, but this in most of the data analysis, you didn't do that. But sometimes you can do it with data too. You shuffle your data and break one thing apart. Or if you do PCA with this step, you take out one step at a time and see which step is contributing. So that involves a lot of back and forth. Okay. And PCA is an unbiased method, meaning you don't have any knowledge. You don't have any knowledge about the cell structure, the tissue you're working with, or the function of any genes. It's just numbers in the table, right? And then, so sometimes there's a good side, it's unbiased. There's also a downside. You can only do so much with unbiased methods, right? Okay, so UMAP is an alternative to PCA. It's also dimensionality reduction, similarly working in data in a table fashion without any more knowledge about what genes are doing, but it's more complex than PCA. PCA runs very fast. You just saw we had 40,000 cells in 400 dimension. In seconds, it reduced it down to 20 dimension because people have generated very efficient algorithm to do PCA, and it's all just linear algebra. But UMAP involves a lot of calculation, and it cannot be explained simply by a rotation or anything. But what it does is it tries to preserve local structure or your neighborhood, if there's any, as much as possible while you reduce it down to a lower dimension. What does it mean? It means if you guys are sitting here, you know who your neighbors are right now, right? If I were to squash you into a plane or into a line, your material, your neighbor are still close to you. That's what it tries to do. But it tries to stretch you in different ways, you know, and the global structure will no longer be preserved. For example, I'm now sitting opposite to, I don't know, you sitting in the back. But my neighbor is with these two. After UMAP, I will no longer be opposite with him, but my neighbor will still try to be preserved. Maybe I will be still close to the microphone, but there's no way I'm still close to, you know, being opposite to someone. This global notion of distance and direction is no longer preserved. That's a necessary step. Okay. Without going into detail of math, how it works, how can we understand if you are presented with such a thing, right? You could always test it. Although we don't understand everything behind the scene, we could still test it with a toy model and see what it does to our toy model, right? So I just applied this ring data to PCA and show you PCA is just a rotation. It preserves all the structure. What does UMAP do? I wanted to use this ring data and let UMAP to do dimensionality reduction and see and plot the UMAP trajectory. And if I did that, I will get something like this. You see a lot of wiggles and breaking point a little bit. But what it does is actually is it's a complex stretching and doing, but at the end of the day, they just try to preserve the local structure. So points that are neighboring to each other are still neighboring to each other after UMAP. And so yeah, that's UMAP. And then what does it do to the actual data though? So this is what PCA does to the actual data. This is what UMAP does to the actual data. PCA try to rotate your cells in a way to reveal the most compelling pattern, maybe the major cell types. But UMAP will reveal more structures, but such that cells in the neighborhood are similar to each other. So then you will see that more cells are reviewed this way, but maybe that wasn't reviewed in PCA in the first two PCs. For example, this group now correspond to this group now lives on their own. But among these branch, there's a few different things actually separate from each other. that can be reviewed by UMAP. That's why it's powerful. But keep in mind the global structure cannot be trusted, as the example shown here. Points, for example, that are polar opposite to each other in the PCA will preserve it, right? And then UMAP, these wiggling is just nonsense. It just displays things in a thing, and but you just keep in mind you can only interpret the neighbors in UMAP. Yes. Okay, so I'll show you how to do UMAP on this ring data again, and you'll apply it to UMAP, and that's it for the day. It turns out coding wise is exactly the same. Even the syntax is similar. So we already did most of the groundwork. Here we applied PCA to the ring data. Now I just want to slightly modify it. So here's how I did PCA. I copied it over, and now I wanted to do UMAP. It turns out people have designed the code such that you can use it quite conveniently in the similar way as in PCA. So rather than say I want to do PCA, I just say I want to do UMAP. That's it. Why is that? Because I imported a package called UMAP here up front, right? Here I installed a package called UMAP learn, and I call it UMAP. Within the package, there's a function called UMAP, capitalized. So I would just use, and it turned out to have the same syntax. UMAP, I wanted to reduce the components to 2 also. So rather than call them PCA, I'll call it UMAP. I guess I'll call it UCs, perhaps. And then again, you apply fit transform on the original data X. And the results, I will call it X UMAP rather than X PCA. Then check shape. Oh, now, by the way, now you see that it takes quite a while because it's doing a lot of computation. Okay, after 21 seconds, it's done. And then I wanted to plot the result in a similar way. Rather, I would call it UMAP, right? I just renamed it X UMAP and send X UMAP in, and so on. UMAP transformed ring data, and then you see now it looks like this. It's no longer a ring, it's kind of a ring, but actually if you run at different times, it'll give you a different shape. But how do we check whether the neighbors are still preserved? We can color, we can try to color it by the original angle called theta. You still remember going backwards. Let's try to, there's no theta. Let me, is there a theta at all? So let me go back and see. Yeah, there is random theta, and maybe it's called confused. Yeah, there is an array called theta. I see. The complaint, they said, is you supply a C argument or a color, but not both. So C and color cannot be in the same line. Here I have C, but I also have color. Let me just remove that color. I didn't want to have that. It's just AI added that part. So and C. Okay, so now it works, right? So you see the neighboring patterns are still preserved because dots with similar color are still close together in UMAP shape. Yeah, that's how you do UMAP, and then you just need to apply it on the real data. Okay, give you 10 minutes, and you can share your results, ask me questions, and that's it for the class.
Yeah. No.
No, because this like the function is defining a new object you want to use it to. This is not actually the data. The syntax is a little using, but this UMAP calls for that function, and then here you specify how many components. So yeah, they want to store
Oh,
all
so
that
this is just the function.
Yeah, it's
Oh,
just
I
a
see.
function.
Oh, okay, so the
This
PCS
function-
had data.
Yes,
Is using
yeah,
it.
exactly.
Okay,
So-
then, right, look.
Yeah, you can do PC
Oh, okay.
because PCS is actually
Still
has the
function.
data. Yeah.
Okay, gotcha.
Yeah, you could check whether, but it's also a great reminder. I probably set up a bad example. You really want to have similar things be named. Similar. In this case, PCS is actually your data.
Oh, I see.
UCS is actually just an argument. It's a function you want to use.
It's just applying
Yeah.
two components to that.
Right.
Okay.
So usually we shouldn't name it. Actually, I should call this some UMAP function, but just to make yourself, because initially you figure out, then you become unconscious and later, yeah. It happens quite often with usually the best practice is when you define it and you try to figure out what it is, and then don't mess up later. Yeah, so I did it. Yeah, but yeah, sorry.
Thank you, Dad. Thank you.
Oh. Did I mess up with the projection? Ooh. Okay, so let me just show you one last bit, which is if you rerun UMAP every time you'll get a slightly different shape, and that's okay. Okay, then for example, here I rerun it and replot it. Now it's a V shape. Now if I rerun it again and replot it again, now it's a it's a different U shape or whatever. But you see, it's gonna be different. The shapes doesn't make much sense at all. The what they try to preserve is just the neighborhood. Neighborhood always stays together. Okay. Okay, that's so much for today. I'm going to stay here if you have questions. Otherwise, I'll see you tomorrow.
Can you just show you? So I applied, I don't know if this is correct.
Yeah.
The kind of the PCA, I don't know if
Yeah.
this can be used.
Yeah, I think so.
And then,
Actually, it ran through his.
yeah, it did. I just don't know if it makes sense. And then if I did that, what about these ones? How
You
could
want-
I fill,
Okay.
how could I, fill these? So
Okay.
what are,
Yeah. It's just a selection problem. So this usually has two columns. Here I want to just select the first column and send it in here. Select
Yeah.
the second column. So yeah, the way
In
we
parentheses, oh,
yeah,
okay.
it's
Oh,
a
okay.
square bracket.
Yeah, because I was gonna just use the square brackets, but I didn't
Yeah, yeah.
understand. I have to repeat.
And
Oh, okay,
and
okay,
the
okay.
syntax is a little weird actually. So you first within the square bracket, they wanted to spec like an Excel sheet. They wanted to select first the rows. So you want to select all the rows. The way to do that is just to put in these and then
Comma,
comma,
zero.
0.
Yeah.
They start from 0. Yeah.
And this is the
Yeah, yeah.
comma one.
Right.
Yeah.
Right.
Yeah, because I forgot the UCS and it had a
Yes,
it had
because
a
you select
red
from-
line underneath and I'm like, yeah, I just forgot to add those.
I see.
And then this is now running because I just copied your
I see.
your
Yeah, because you see usually there's one line that's heavy lifting. It's this one. You see fit transform takes quite a long time.
Yeah.
So yeah. So
But I could use that one and that one too.
because it's because it's still running. Let me try to kill this.
Okay.
So we can run that. Oh, now
It might be because it was trying to run the other one.
Right. So let's try if
That took 4 minutes or so.
okay. Yeah, that's the problem when debugging takes troubleshooting takes long.
Yeah.
I guess I don't know how experiment.
But I
you always figure out
try to run it after.
Or actually, we usually usually we are impatient and it's always good to try to run it on a smaller data.
Yeah.
Okay. So let's just instead of running it on say 80,000 cells, let's just run it on the first 100 cells.
Right.
So that should be done in a few seconds. Yeah, 4 seconds. Okay, then we can see if it goes through. It still doesn't.
Yeah.
So that's because I subsetted the cells, but the coloring was not. So the color by gene expression, so UC only have 100 cells, but this still has the original number of cells. So let me just take the first 100.
Okay.
So it went through. It's extremely small because but now
I get the idea.
It went through completely, right? Now you can say, do we just to make sure let's run it on 1,000 cells. If it walks through, you can remove that, right?
Yeah.
Yeah.
Okay, thank you.
Yeah. Oh, now it's okay.
It's okay. I'll just remove the reduction.
Yeah.
Thanks.
Hi, Pingming.
Yes.
I had a question. So my UMaps, I put them on the Google.
Okay.
I think I'm slide 5.
So slide 5. Yes.
Yeah. So I when I initially tried to try to run the code myself, I got the map on the left, but then when I followed along with yours, I got the one on the right.
You mean this version?
Yeah, I'm not sure what was completely different, but it basically looks rotated, and I'm not sure what would cause that. Do you have an idea of what I may have done?
There's a random initiation steps in UMap, I guess in many parts of UMap. So
okay.
Yeah, the rotation could be could be because of that. I don't know exactly what's under the hood, but yeah.
Would that change the meaning of the data?
Not at all. Yeah, it's it's even I wouldn't trust the shape of it. What it tell you is you have a few islands or neighborhoods in this data that seem just certain groups of cells are similar to each other than others. Then that's it. Yeah, the shapes are you can trick UMap into producing many different shapes as you like. Without and they all work equally. By UMap standard, they all reduce dimensionality equally well. definitely.
I see. So basically the clusters of cells that are similar to each other will be mapped near each other no matter what iteration of map.
Yeah,
Okay.
that seems to be stable. Although, yeah, that seems to be stable here, and I believe it's yeah.
Okay. I was just trying to figure out what I had done to change the visualization and I couldn't figure it out. But thank you for explaining that.
Yeah, no worry. So think of it as UMAP is trying to find a solution that has many answers equally well answered to the same question.
Yeah.
Or thinking about a ball rolling down um, along a very complex landscape, and there are many valleys in this landscape. Any one of them are equally well. And then depending on where your ball is started, it'll roll down into different valleys. And where you started is a random thing. It's a random variable in UMAP. And then they just optimize towards
Sorry. So is that why when you reran your own code and it looked more V-shaped and more U-shaped, that was that change? Okay.
yeah, that level of variation is intrinsically there in UMAP. So any time you run it, it'll be a different answer. There are ways you can for reproducibility though, you could try to make it fixed every time you run it by specifying a fixed random initiation. That I think you can do. Maybe ask AI to do it for
Yeah.
reproducibility purpose, but still, the pattern, the specific shape doesn't mean anything.
It's just the cluster, like where the individual cells are mapping to.
Right,
Okay.
right, right.
All right. Thank you very much.
Yeah. Okay, yeah.
Quick question, sort of going back to the PCs.
Yeah.
So are we running a PCA to then inform the UMAP?
Yeah, you can do that. That's actually what people usually do.
Oh, I
Because
see. I
one
missed that.
thing
Okay.
about UMAP is too slow
Yeah.
and too many dimensions, and
I see. So you reduce it first and then run
Right,
the
right.
UMAP on the
like,
reduced.
yeah, you
Okay.
produce a recipe that's both efficient and powerful, kind of like
Okay.
that. Yeah.
Okay, gotcha. Okay, just making sure that that's what we're doing. Thank you.
Question.
Yeah.
I have no idea why like these one you said.
Okay. Not module object is not callable.
Yeah.
Um,
Is it the top of the song or something? I think more complicated than that.
hmm. Okay, one thing is to just first locate the error. I believe error is over here. So if we remove it, so I guess, yeah, if you're gonna run first line, obviously it doesn't,
Is the other?
because it has no shape.
Yeah.
So what is UMAP in your case? Let's see. Okay, it is a function that you can call.
Mm-hmm.
So yeah, it has neighbors and etc.
Yeah.
But do I have any components close to? Yeah, it seems to be okay. So it looks like this line, I was wrong. This line should be able to run too.
Okay.
Let's see. Yeah, it did. So
Mm-hmm.
yeah, first is to locate, it must be this line. UCA fit transform. So we know UCA is correct. It has a function called fit transform. How about we just reproduce it here? So UCA equals this, and UCA dot fit transform. What I was worried, yeah, it has it, it recognizes it. Yeah, it's a function. It's on encoding X. So it must be this features it's complaining. So if I do this, do I reproduce the error? Looks like it's running. So Again, because it's surrounding, it takes a long time because your feature matrix is too large,
Mhm.
right? Usually what we do is just first save your time to make
Mhm.
sure, just select the first 100 rows.
Okay.
It should run faster. Let me interrupt it.
Mm.
Okay, and then run it again. Hopefully it runs through in seconds. Ah, yeah, it does run through 7 seconds.
Mhm.
Yeah, so it looks like it's working. And I guess, yeah, so this line should work through
Okay.
theory. And then, yeah, I don't see a reason why this wouldn't work, right? Just to make sure here, because it's still too much. So maybe I also select the first 10 rows,
Mhm.
10 columns, just to make sure it runs even.
Sweet.
Because it doesn't like me selecting things this way. That's a
Mhm.
peculiar thing. If I do this, it'll run through in a second. As features is a table, it's not exactly a matrix. If I do dot values, it will turn it into a matrix, and you can select it this way.
Okay.
Anyway, that's a detail you can learn from. I just wanted to use a small table to run this through in 0 seconds.
Okay.
Then yeah, just see
Get
if
the
it-
shape.
the shape, okay. Oh, that's because, I didn't do UC's equal this, so I should do UC's equal, just do exactly the same thing. Yeah,
Okay, 10 2. Okay. 802. Okay.
yeah, 102,
And
so let's try 1,000 style, does it work through? Yeah, 1,002. Looks like
Okay.
it's working. So now you may remove this. So nothing's wrong here. In some case, you said the module is not callable. Yeah. Anyway,
yeah.
so because it's not reproducible, let's
Yeah,
not
that's
worry
fine.
about-
That's fine. Yeah,
But
as long as it's
Yeah,
working and it's
yeah.
working.
But yeah, a good practice is if it's an error, you first reproduce the error, then you locate the error on which line, right? Then you try to figure. So like it's like peel off an onion thing, right?
Mm-hmm. Yeah, and because my lab mate is defending tomorrow.
Yeah, no worries.
But I mean, like, can I join on the Zoom?
Yes. Do you have send me an email? I'll give you a Zoom link.
Okay, okay. I think I already
Yeah.
sent you an email.
Oh, really?
Today at 4 a.m.
Oh, okay.
Yeah.
Okay, let me see.
Yeah, because yeah, he wants me to be there helping him setting up stuff, but I can still join on the Zoom
Okay.
when he is defending his thing.
Just want to double check if I got it.
Elizabeth. This one, this is my email.
Got it.
Yeah.
Send it here.
Okay.
Yeah.
Okay.
Sorry, let me just end meeting for